We're all set, right? Alright. Now time.
It's going to be good. Okay.
I don't do shit lectures. All right, guys, can we get started now? All right, Immunology Microbiology. This is 30 marks of your 100-mark exam. In other words, 1/3 of your entire exam, and I have 1.5 hours to present it. There's 87 slides, so it's like a minute a slide. All right, long story short, a lot of the microbiology is super text heavy, and I'm going to skip over a couple of the slides. On there, I literally have like a list of the bacteria or fungi or whatever that you should just go learn. There's no point in me reading the slide to you. The lecturers do that anyway. All right, so let's get started. This is what I'm going to cover for the immune system. All right, immune system. There's two halves to it. The innate immunity and adaptive immunity. The innate is the non-specific one. It's the one that reacts the same to every sort of pathogen, and it provides an initial defense. It's the one that also initiates the adaptive immunity to come into play. The adaptive immunity is the specific one. It's the one with the memory, and it's the one that really plays probably the larger role. Just a picture. All right, cells of the immune system. So you've all probably seen this. That's just showing you all the different cells and where they come from. They all start off in the bone marrow, and then they split. They either split into myeloid, into myeloid or lymphoid cells. So the myeloid ones include neutrophils, macrophages, basophils, eosinophils, and mast cells. While lymphoid cells are your B cells, your T cells, and also your NK cells, as well as innate lymphoid cells. No one cares about those. And then there's the dendritic cell. These guys are the only ones that derive from both lines. So they come from myeloid and lymphoid derivatives. All right, so the development of lymphocytes. Lymphocytes refers to the B and T cells, mostly, at least. Your B and T cells both come from the bone marrow, but they mature in different places. B cells mature in the bone marrow, B for bone. T cells mature in the thymus, T for thymus, easy. All right, and so whilst they're maturing, they actually undergo a screening process. Yeah, I don't have time to pause, sorry. All right, and this is what the screening process looks like. What happens is, at the very start in the bone marrow of your lymphoid progenitor, they become pro B or T. Pro anything basically means it hasn't arrived at its final stage yet. It starts building part of its antigen receptor, one half of it. If it fails to do that, it gets killed. If it succeeds, it moves on. Then it builds a full receptor. If it fails that, it gets killed. If it doesn't, it moves on. And then there's the final part. This is the most important. This is what prevents autoimmunity. Top is showing weak antigen recognition. What that means is that the receptor doesn't look at antigens that are normal inside the body and think that they're foreign, which means they're not self-reactive, which means that's good. So they're positively selected for. Whereas if you have an antigen that a receptor that is too self-reactive, in other words, it's too good at its job and it reacts to everything inside your own body, that will cause autoimmune issues if it were to live. So it gets killed. That's called negative selection. It's a bit more specific for T cells. They're meant to react to particular MHC class markers. Your MHC class 2 and MHC class 1, CD8 for class 1, CD4 for class 2. Weak recognition is what's necessary. If it strongly recognizes either one of those MHC markers, it will result in autoimmunity as well, which we want to avoid. All right, and this is just a summary of how those lymphocytes, where they develop and mature. Start off as bone marrow, become either one, mature in the bone marrow for B cells or mature in the thymus for T cells. Then they move on. They go through the blood and lymphatic system to arrive at wherever they want to stay. That can be either B cells sometimes stay in just the bloodstream and just keep going around. T cells and B cells like to go to lymph nodes and stay there until they get activated. And there are also other lymphoid tissues, which we'll get to in a moment. This is just a nice picture of all your myeloid cells. I suggest you recognize what a neutrophil looks like. Tushar talked about it before. It's got a lot of granules in it. And also recognize what your dendritic cell looks like, which is pretty obvious because it's the only one that looks like a star. Your eosinophils are important for attacking your parasites. They're small, they're incredibly tiny, but they attack in numbers. They literally all, like if there's one parasite, they'll all go onto it and stick to it until that parasite dies. Your basophils, they're involved with allergic reactions, and if they get activated, they can cause really severe tissue damage as well though. Mast cells, if you've ever done VC bio, you would know, they're the ones that cause your allergic reactions. So if anyone's allergic to nuts or egg or whatever, they're the kind that cause the airway to constrict and people to die. All right, macrophages. They're the ones you've probably all heard about before. When they're in the blood, they're called monocytes. Until they reach the tissue, they're not called. So when they reach tissue, they're called macrophages. There's two types of macrophages. They're either fixed or they're free. Fixed ones are permanently residing in that particular tissue. For example, alveolar macrophages. Your alveoli are in your lungs. Those alveolar macrophages never leave your lung. Your osteoclasts never leave the bone because they don't have a function anywhere else. And then there's the free ones. They're the ones that literally go as monocytes in your bloodstream, and then they reach a particular tissue. They migrate through the endothelial cell wall, so your vessel cells, your vessel, and then into the tissue. The function of the macrophage, you can read that yourself, but basically they engulf the pathogen and present it. Then there's neutrophils. They're the most abundant white blood cell in your body. They're also possibly pretty much the first cell that will defend you against everything. What they do is that they proliferate crazy, like heaps of them, and they'll just build up in the area of infection. And that's why if you have like a really bad infection and if you ever poke it, like pus comes out, that's basically dead neutrophils. What a neutrophil does is it can either phagocytose or it does a thing called netosis. Currently, it's not super well understood, but netosis is basically where the cell erupts and it will shoot out a net of its own DNA to capture the bacteria. That's the neutrophil, that's the net, that's the bacteria. If you're interested, there's kind of like some cool videos, but yeah, no time. Dendritic cells, they're the ones that look like stars, and they're the main and the most important antigen presenting cell because they're the main link between the innate and adaptive immune system. We'll come back to more detail in a moment. Okay, so lymphoid organs and tissues. Lymphoid organs include your spleen and, well, basically anything that's not a vessel is considered like an organ. Your lymphatic vessels are the lymphoid tissue. Lymphatics is like a drainage system. It's literally like the sewage pipes that run through your body. What happens is your vessels, Tushar talked about this, your blood will leak out and into your tissue. If nothing ever drained away that fluid, you will literally bulge up until you look like Augustus Gloop. And what happens is your lymphatics will take in that fluid again and drain it back into the body and back into the bloodstream. And so what your lymphatics have, they also have lymph nodes. I can't see where they are. There's some of them. Lymph nodes is where your B and T cells sit. And so all your lymph will run through those nodes until it reaches the bloodstream. Your spleen is also super important. That's responsible for detecting any antigens that's in your blood. This is a lymph node. It looks like a bean. Important parts of it are the afferent lymphatic vessel. So afferent for whatever Tushar said, approach, I don't know, arrive. That's where your dendritic cell comes into your lymph node. This is not labeled, but that's the efferent lymphatic vessel. And that's where your activated T and B cells will leave the lymph node. Your B and T cells have particular spots inside the lymph node, and that's controlled by particular cytokines. So there are cytokines inside the lymph node, and they congregate in specific areas. The ones that attract B cells are around the lymphoid follicles. So that's on like the somewhat outside edge of the lymph node. And then in the middle is called the paracortex, and that's where your T cells generally sit. So when a dendritic cell has engulfed a pathogen, it'll come through there, arrive, activate a T cell, and leave. Your B and T cells, like from the very start, how do they arrive in the lymph node? They don't come via lymphatics. They come via a vessel called the high endothelial venule. Fancy name, doesn't have anything special. And that's just another picture. Goes in via those lymphocytes, enter via the artery. Your dendritic cell presents, they exit via the efferent lymphatic and into your bloodstream. When they get to the bloodstream, it will float around your body until it reaches that site of infection. All right, the innate immune system. Non-specific, as I already said, and it has 2 lines of defenses. In year 12, you generally learn first, second, and third line of defense. So the first and second qualify under the innate immune system. So this is just separating into 3 categories. Your physical, there's your skin, and it has to be intact. Skin with like abrasions, wounds in it is useless because bacteria will get through it. So it has to be intact skin. Mucous membranes, so the stuff like up your nose, through your gut, that's all mucous membranes, and your cilia. They're these little hair-like things that beat like very repeatedly in your airways to get rid of mucus and anything you breathe in. That's why your lungs basically have nothing, like they're sterile, because your cilia prevent anything from getting in. Then there's chemicals. Stomach acid. Low pH means that nothing can survive there. And then there's saliva, tears, urine, and sweat. Saliva have enzymes. Tears also have enzymes that will kill bacteria. Urine is more of like a mechanical thing. When it comes out so quickly that it will wash off the walls that it passes through. So any bacteria that are in your urethra will get like pushed off because of the urine. And then sweat. Sweat is super salty. So think back to Chris Wright, lecture 1 of the year, talking about osmotic pressures. If there's heaps of salt outside the bacteria and not as much inside, it will literally shrivel, like it will pull the water out of the bacteria until it shrivels up and dies. And then there's histamine and interferons, and complement, and we'll go through those in a moment. Cellular, neutrophils, which we've talked about, macrophages, natural killer cells, and also technically a natural flora countercellular. They sit on the outside of all your skin. All right, complements. This, if you have time, go for your life, go and learn it. I'm not going to go through it because there was 1 question on it and you can literally guess your way to getting it right because all the other options are so wrong that you can't get it wrong. Okay, so what happens is, I'll give you a clue. I'm not allowed to say the question, but I'll hint at what it is. All right. Yeah. So complement. These are proteins that are always in your bloodstream. They float around until they get activated. Once one gets activated, it will literally cause a series of events to occur. There's 3 pathways. You've probably seen this or this picture. The same thing. This one is the one they like to use. So there's the classical pathway, and that's just basically your complement protein binding to a pathogen and then all the antibodies on a pathogen and initiating the adaptive immune system. There's the lectin pathway, which is, lectin is a protein. It binds to the pathogen surface. It initiates the complement proteins to work. And then there's the alternative pathway, and it's not understood, and I also have no idea what that says. Like, it makes 0 sense to me. But all in all you really need to know is that it leads to 3 outcomes. These 3 outcomes. Recruitment of immune cells. In other words, it will induce inflammation. The more immune cells there are, the more likely you're going to get rid of whatever's infecting you. Increased phagocytosis, once again, to eliminate the microbe. And then the last one is the cool one. It's called formation of a pore. The pore is called a membrane attack complex. It's this thing here. What happens is C5b, C6, C7, C8, and C9. So everything from C5b onwards will join together to make a hole in the cell membrane. If there's a hole in your cell membrane, everything's just going to leak out and the cell is going to die. In other words, whatever that bacteria or whatever it was is just going to die. So remember your C5b onwards is what makes your membrane attack complex. Do I need to repeat that? No. All right. That's just another picture if you prefer. Some people, there's a lot of controversy as to whether these 3 outcomes are separate pathways or whether they're all actually linked up together. It doesn't matter. They all operate the same way. All right. PAMPs and pattern recognition receptors. They're just really long words for actually really simple things. PAMPs is anything that is recognized as foreign in your body. It can be something on top of a bacteria, like a lipopolysaccharide. That's what LPS stands for. Or it can just be like sugar, glycoproteins. It can be the RNA in your viruses. Anything that is recognized as foreign. Your pattern recognition receptors are what detects those PAMPs, what detects the fact that there is something foreign in your body. There's 3 main types of receptors you need to know. Toll-like receptors, nod-like receptors, and rig-like receptors. Your toll-like receptors are the most important ones. So they can be seen on 2 places in the cell, either on the external cell membrane, so on the very outside, or on endosomal membranes. Endosomal means just basically anything that's membrane membrane bound inside the cell. And they just detect different things. So extracellularly, they detect bacteria because bacteria are generally outside the cell. And then the endosomal ones, which is intracellular, will detect the virus because viruses infect inside the cell. It's all quite logical. And then there's these ones. So nod-like detect bacterial cell walls inside the cell, and rig-like receptors detect viruses. The reason why nod-like bacteria can detect bacterial cell wall is when it's been engulfed or something. It's not like somehow the receptor has touched a bacterial wall without leaving the cell. That's not possible. The most important kind, as I said, is the toll-like receptor, and this is what happens. There's like 9 different types of toll-like receptors and probably more, and they all detect different things. The one that your lecturer loves the most happens to be TLR4, and that detects lipopolysaccharides. Lipopolysaccharides, doesn't matter. When they bind, it causes signal transduction. Signal transduction basically means one chemical will signal another, and it'll keep going until its final effect has been done. So the first thing, signal transduction to these adapter proteins. Adapter proteins will activate transcription factors. Think back to the French dude who lectured you. Transcription factors are things that promote or inhibit your transcription. In other words, it works with your DNA. So there's 2 possible kinds, NF-kappa B or interferon regulatory factors. They go into the nucleus, they do something to the DNA to cause transcription, and it will result in production of molecules. The important one that we need to remember is interferon, type 1 interferons, which includes alpha and beta. What they do is they create the antiviral state, and we'll go into that in more detail in a moment. But they can also produce cytokines, which basically induce inflammation. It activates the adaptive immune system, and yeah. That's inflammation. You guys have had tutorials and lectures on this. I'm not going to go into it in detail, but basically inflammation, there's the 5 signs. Remember the 5 signs, and basically everything else fits together around those 5 signs. So when bacteria comes in, it will cause macrophages to release cytokines and chemokines. Those cytokines and chemokines will cause vasodilation and increase vascular permeability, which in turn causes the redness, the heat, and the swelling. The inflammatory cells, and all the cytokines and all your macrophages and all the immune cells in there, will further release more chemicals called inflammatory mediators. Inflammatory mediators will act on your nerves inside the site of infection and cause pain. And whenever there's swelling, pain, heat, and redness, you probably don't want to move that part of your body, so there's loss of function. Do you guys know what the difference is between a cytokine and a chemokine? No? Basically the same thing. A chemokine is a cytokine, but a cytokine isn't a chemokine. All right, here's a definition. A cytokine is a protein that affects behavior of cells. A protein that affects behavior of cells, okay? And a chemokine is a type of cytokine that attracts neutrophils and monocytes. So a chemokine is a cytokine that attracts neutrophils and monocytes. But generally in the exam and just in life in general, they like to swap between the words and not use them accurately. It doesn't really matter. They're basically signaling molecules that cause inflammation. All right, how does inflammation start? You have an inflammasome that needs to be activated. This is your inflammasome. It looks really complicated, but really you only need to remember one thing. You don't want inflammation to happen easily in the body. Otherwise, you're going to be permanently red, swollen, and hot. Yeah. High temperature. Yeah, anyway. Okay, so for the inflammasome to be activated, there are 2 pathways that have to occur before it can. So the first one is your NLRP3. It's a sensor. It gets stimulated, and it will join with these other 2 components to form this inflammasome. At the same time, your cell, if it's come across a PAMP of any sort, it will induce synthesis of pro-interleukin-1 beta. Pro means it's inactive. So for that to become activated, caspase 1 from that inflammasome has to come off. That will induce pro-interleukin-1 beta to become interleukin-1 beta. Interleukin-1 beta is the very important cytokine that will go out and basically induce inflammation. Macrophages. They can be activated in a variety of ways. You can read that yourself. Types of macrophages. All right. There's two, like we tend to tell you, macrophages cause inflammation. But there's actually another type of macrophage that prevents inflammation. So you can see here, generally any like microbial insult, so just any pathogen that comes in, and also particular cytokines like interferon gamma. Remember interferon gamma. Remember what it does. Just remember it. It will induce the macrophage to become the M1 type, so classically activated. This M1 type of macrophage will cause inflammation. However, if you have interleukin 4 or interleukin 13 or 14, that will induce the alternatively activated macrophages. They like to do things differently, and they are anti-inflammatory. They basically come around at the end of an infection, and they want to fix everything up, because otherwise there's nothing that's going to stop the inflammation. Natural killer cells. So these are the only innate immune cells that are considered lymphocytes. Just a technicality, and also how they're formed. But natural killer cells what they do is that they can detect the difference between self and non-self via the MHC1 marker. So this is what it looks like. In a self cell, your MHC1 marker will be made correctly. There'll be an activating part of it, and there'll be an inhibitory part of it. The inhibitory part, if bound to the natural killer cell, tells the natural killer cell that this is a self cell, that you shouldn't kill it, and so no killing happens, which is normal. If, however, you have a virus or perhaps a cancer cell, they tend to not be able to express their MHC1 markers correctly. In other words, there is no inhibitory section, which means this inhibitory receptor doesn't get activated, which in other words, killing will happen. All right, and interferons. These are specifically interferon alpha and beta. They're released by virally infected cells. They do two things. They activate NK cells to kill the infected cells, and then they prevent non-infected cells from becoming infected. They do that by interfering with viral replication. Most of the time what that means is they basically make the cell membrane, they change it in such a way that the virus can't get into the cell. All right, adaptive immune system. A lot of terms. The only one that's probably harder is epitope. Basically means it's just the part of an antigen that B or T cells receptors can bind onto. And please don't do what all the year 12s like to do. Don't mix up antigen and antibody. It's stupid. All right. How do you tell the difference between self and non-self? First of all, there's receptors. Your B cell receptors, in other words, your antibodies, also known as immunoglobulins, or just written as Ig, they are normally on your B cell, but they can be secreted. And they can bind to the entire antigen or just a part of it. Whereas T cell receptors, they only react to linear epitopes. In other words, they only react to pathogen that has been engulfed by a dendritic cell and is now presented on an MHC marker. And these receptors can't be secreted. These receptors have an alpha and beta chain, doesn't really matter, but that's extra information. All right, and now there's MHC markers. The difference between these. Class 1, they activate CD8 cells, as in the cytotoxic T cells. MHC class 1 are present on all nucleated cells. So your red blood cells don't have them, but they're still considered self, but every other cell that has a nucleus will have MHC class 1. It presents antigens from viruses. This is called the rule of 8. In other words, 8 as in CD8 and 1 for MHC class 1. 8 times 1 gives you 8. In the case of MHC class 2, you have class 2 that affects CD4 T cells, so 4 times 2 gives you 8, if you ever struggle to remember which is which. So CD4 as in T helper cells react to MHC class 2 markers that are on antigen-presenting cells. APC stands for antigen-presenting cell. All right. How much detail did your lecturer go through this in? Because it was on the slides, but it's like the stupidest thing ever. All right, I'll go through in like 3 sentences. Basically, your receptors are super specific. For them to achieve this specificity, it means that DNA has to be shuffled around to make those different receptors. There are things called DNA cassettes. Think about it like a cassette tape. They're basically, all that set of music is on that one tape. Like a DNA cassette, all that chunk of DNA, the important stuff is in that section. And so those cassettes can get shuffled around and put in different orders to make different receptors that all have similar functions, but it's specific to different antigens. There's a C-mu, which is the constant region, and we'll go through that later. And so basically, in your B-cell receptor heavy chain and your TCR beta chain, they incorporate V, D, and J DNA cassettes, whereas your BCL light chain and the alpha chain only have V and J. If you have time, you can go through this in detail, but all it basically says is that, okay, there's an infection, we need to create receptors. Your DNA is going to shuffle itself around to create the receptor necessary and spit it out. That's it. All right. Now, there's 2 pathways in the adaptive immune system, humoral response and the cell-mediated response. What we're going to do now is we're going to swap to drawing. Let's do this. This is what we're going to write out. But pretty much once you understand this, you will basically understand the whole adaptive immune system. So now I'm just going to run through it slowly so you guys can draw it whilst I go through it. Do you guys all have paper, or at least pretend to have paper? Surely you guys got enough free pens from those insurers to be able to have a pen. All right, let's get started. Okay, first of all, we're going to start off with an antigen. My year 12 English teacher hated my handwriting, so I apologize, but I haven't gotten any better since. All right, we have an antigen, and there's multiple things that it can do in the body. First of all, is that it tends to be engulfed by the innate immune cells. Well, we're going to say it's been engulfed by a macrophage or a dendritic cell. Yep. So once it's been engulfed by that cell, the cell will then process that antigen and display it on the MHC class 2 markers. And then that macrophage or dendritic cell technically becomes an antigen-presenting cell. Because before that, they have no antigen to present, so it wasn't an antigen-presenting cell. Now it technically becomes an antigen-presenting cell. This antigen-presenting cell will flow around the bloodstream and the lymph until it reaches a lymph node. So, when it reaches the lymph node, it's going to stimulate. It will stimulate a T helper cell, a naive T helper cell, which once stimulated becomes an active T helper cell. This T helper cell then can do a lot of things. First of all, it will stimulate itself to undergo clonal expansion and differentiation. So, clonal expansion and differentiation. Yep. Do you guys all know what that means? Yes? No? Yeah. So clonal expansion is basically where your cell makes copies of itself, and differentiation is where it gets a proper role. So what that actually means is that the T helper cell will make more T helper cells, but it will also make memory T helper cells. And now that T helper cell on its own doesn't do much. So depending on the type of antigen, there'll be two possible pathways. We'll look at the humoral response first. This is when the antigen is an extracellular antigen. So for example, let's say bacteria comes in. What can actually happen is that it can on its own, the free antigen can activate a B cell. So your B cell. But on top of that, your T helper cell, once it's been activated, can also stimulate the B cell. Your B cell then will undergo clonal expansion and differentiation to create two types of cells. Which two types? Yeah, you can also read it if you have good eyesight, just. All right. So it will create active B cells. Active B cells are also known as plasma cells, which I heard you say. And they'll also produce memory B cells. All right. Clonal expansion plus differentiation. Your active B cells have an important function, which is to produce and secrete antibodies. What these antibodies do, we'll go through in more detail in a moment, but in a few words, it defends against extracellular pathogens by binding to the antigen and therefore making them easier targets. So I don't know if you've ever seen that meme where it's like eat more cake and you'll be harder to kidnap or something. If you're fatter, if you have heaps of antibodies stuck on you, it will be harder for that bacteria to get away. So just think about fat people not being able to run fast. That is literally how your antibodies work. Yeah, sorry. Easier targets for the complement system. And phagocytes. I will be uploading a copy of this later, so don't freak if you can't get it down, but that's general gist. All right. That is the humoral response. Humoral response basically means there will be antibodies that are produced. Have you got most of that down? Yeah?
I think it's a distinction. I think isn't it like the dendritic cells are only ones that can activate the T helper one?
So the thing is, how do I explain this? Some people believe macrophages can activate it, and some people also think macrophages can activate B cells as well, but it's all still currently in research. As in the textbook literally says it's unclear, like dendritic cells definitely activate T helper cells, but macrophages may also potentially play a role in it. And so, I don't know, up to you, but dendritic cells is more accurate, macrophages may come into play. And in terms of like the exam, they do swap between the two because they are both technically antigen presenting cells. Yeah? All right. That's your humoral response. We're going to move on to the other side, which is called the cell-mediated. Cell-mediated response. First of all, what happens is your activated T helper cell will go back and induce the macrophage or dendritic cell to kill whatever it's engulfed. So it will release cytokines to stimulate the phagocyte to kill anything that it's engulfed, so the engulfed pathogen. Is that easy to read, or do I need to zoom in? Yeah, okay, I didn't hear that, all right. Zoom? All right, this looks hideous. All right, then what this T helper cell can also do is that it can activate cytotoxic T cells. Cytotoxic T cells can technically also be activated via another pathway, which is directly from a cell that is virally infected. All right, so that's another way that it can be activated. An activated cytotoxic T cell will do the same thing as all the other ones, which is undergo clonal expansion and differentiation. And that will produce active TC cells and also memory TC cells. Did you guys go through perforin? Heard of perforin? All right, well, that's what your active TC cells do. They produce and secrete perforin. Yep, sorry. There we go. Yep. Clonal expansion and differentiation. I just can't be bothered writing it, but that's yeah. Yeah, Sonny?
So if it was just bacteria, the T cytotoxic cells still be activated?
Because T cytotoxic cells really only work on infected cells by viruses or cancer cells. So this side is like bacteria. This side is virus, mostly. Yeah? And, yep. So I'll write that in detail. So your perforin defends the body against intracellular pathogens and cancer cells by binding to the target cell. Yep. So what perforin does is that it will create a pore, not a pore, it creates a lot of pores, so that granzymes can enter. Granzymes is just a fancy name for enzymes. And what enzymes do is that they'll cause apoptosis. Do we all know what apoptosis is? Yeah. It is the controlled killing of a cell. In other words, the reason why it has to be controlled is if this cell was just burst, all those virus particles will then come out and infect other cells. So it has to be controlled killing, which in other words, those virus particles are taken in and killed off individually so that no other cell nearby will get infected. All right, that's that. Nearly finished. All right, let's say that you're unlucky enough and this same antigen comes around a second time. Second exposure, as we all know, is faster, it's more vigorous, and it basically prevents you from showing any symptoms. More often than not, when you've been infected by one pathogen, the same pathogen twice, you don't even know that it's come around the second time. That's because these three things happen. Your memory TH cells get activated. If it's a bacteria, your memory B cells get activated. If it's a virus, your memory TC cells get activated. On top of that, your memory TH cells will also stimulate those, which creates like a double effect. All in all, that is why a second or subsequent infection by the same pathogen gives you a more vigorous and faster response. That is the end of the story. Are there any questions up till now? Guys? Any questions? All right, so I can't see you. Yep.
Do antibodies help with viruses?
Yes and no. We learn it that they don't, but they do play a role in it, but less. So just take the idea. Antibodies are for your bacterial insults, whereas your cytotoxic cells do your viruses. There is some overlap, but it's just technicalities, and they're never that fussy. Yep. Yes. So yeah, your memory cells will have to undergo clonal expansion and everything. So technically, yeah, I didn't draw an arrow because it makes it super messy, but technically there will be an arrow from here, kind of back to there, and it will undergo clonal expansion, or if it's a TC, it will go back to there and undergo clonal expansion. And that is why after a second infection, you'll get more memory cells and more memory cells. And so like if you've been infected by the same thing so many times, you'll be full of memory cells and you won't actually you won't feel the disease. You won't ever get any symptoms. All right. Yes. I assume it could happen at the same time if you were infected by both things at the same time. Like it's the thing with the immune system is that we like to say it's step by step, but really it's all happening at the same time. At one moment, a TC cell might be getting activated whilst a T helper cell is meeting a new antigen. And it's just all these separate steps could all be happening at the same time. And this is the most logical way to put it out. But yeah, it could happen at the same time. Yeah. Anyone else? Otherwise, you're going to, oh, yeah, go on.
For example, you have like a very minor infection, I don't know, lasts one day and your innate cells deal with it. But then if the dendritic cell gets one of these, would a body still bother to initiate the adaptive immune system?
Okay, so basically what happens is your body technically, like your cells don't really have a brain as such, it doesn't make decisions. So by chance, it will initiate an immune response, but it won't be as big. And it will basically what happens is it will reach if there's like a virus particle, it'll reach a lymph node, your TC cell gets activated, it'll get rid of that particle, it will go through, it will circulate in your body just to make sure there's no other ones, and that's it. Yeah. And that's what, I haven't got it on the actual slide, but this is what this shows. What this shows is that what we just talked about is up to here. Once all the antigen has been eliminated, all the extra cells will undergo apoptosis. Only your memory cells will stay behind for another infection. This is a great picture, but the only issue I have with it is that this timeline is completely wrong. It depends on the type of infection. Different infections have different incubation periods and stuff like that. In other words, this whole process can take really short time or take a really long time. But all in all, this is the general gist of it. All right. Oh yeah, but one thing, clonal expansion. You know, I don't know if you've when you get an infection, you get a swollen lymph node. You might have felt one on yourself. That is purely because your cells have made so many copies of themselves. Do you think about it? When you think about a cell, you think it's so small that if there were 500 of them, it doesn't make a difference, but it does. That's why your lymph nodes swell. And that's because of clonal expansion, because that's how many copies your cells will make. All right, this is just more detail about the humoral response. I won't go into, I'll skip all of that, but going towards the end. IgM, it's a type of antibody, is always the first to be secreted. It's the initial response. It will run around and do the best it can. At that same time, IgG will be slowly being produced by these cells, IgG expressing cells. They're basically isotype switching basically means there's different types of antibodies. Depending on what's come into the body, a particular type will be used. Affinity maturation is where your antibodies are made to be more and more specific for the antigen. And that's using the same concept as your receptor recombination. Your DNA will shuffle itself until it's perfect for that antigen. And once it's realized it's perfect, it's so specific that it will get literally all of them, it will replicate that cell to eliminate the microbe. This is what an antibody looks like. The blue bit here and the red bit here, that's your heavy chain. The green bit here and here, that's your light chain. All four of the chains have a constant section, but it's the tips, so this bit here and this bit here, that vary. That allows for specificity. The important thing to remember though, that this part and this part, if it's on the same antibody, it will be specific for the same thing. It won't have a different shape. It will be specific for the same thing. All right. Types of antibodies. You can kind of read this yourself. Important thing to remember, most important, is IgG is involved with immunity for babies. It's the one that can cross the placenta. It's the one that goes out through breast milk as well. You probably know all of this already, but antibodies can do three things. Neutralize, which in other words, it binds to the antigen and prevents it from becoming toxic or prevents it from causing damage. Then there's opsonization. It's a bit like complement. Your antibodies will bind to your pathogen and make it more attractive for macrophages. Then there's complement activation. Your antibodies will bind to the pathogen and your complement will then bind to the antibody to induce macrophages to engulf it. So in other words, all three of these pathways ultimately lead to ingestion by macrophages. Acquired immunity. We're nearly there, at the end of immunology at least. All right. Yeah. Active, passive. Active is where your body actually makes antibodies. Passive is where you're given antibodies. Simple as that. Natural versus acquired, artificial or induced. Natural means that, it happens without human interference. Induced means humans have interfered and caused shit. So what natural, this is the one that happens on a day-to-day basis. You get infected, you get better. Artificial is like the vaccine. They're purposely infecting you. They're injecting an antigen into you, although it's been reduced in terms of virulence, so it won't cause, you won't feel the symptoms. You're not meant to feel the symptoms. But they are purposely injecting an antigen into your body so that your immune system will respond. Then there's passive. Natural is when, for example, your mother passed on immunity to the child. That immunity will only last a couple of months, and only when they're being breastfed. And that's why it's so important in Africa. Like, no joke, if they don't breastfeed their children, they will have higher rates of dead children if they don't breastfeed them. All right, anyway. And then artificial passive, that's basically antidotes. If you get bitten by a spider or a snake, these are super important. And they're super fast acting, but those antibodies don't last. They will die off and you'll have no memory of it again. So if you get bitten by a snake again, you still need the antidote. I don't think, like, last year our lecturer never covered it. Did they cover hypersensitivities with you guys? No? Thing is, it's like never covered, but it always somehow comes up. So just have a quick read. The only important one you need to know about is type 1 and type 4. Type 1 is the normal allergy. You know when someone eats a nut and they suddenly start choking, can't breathe, and then you need to stick an EpiPen in them? That's anaphylaxis, that's type 1. That's caused by mast cells releasing histamine that cause really severe inflammation. And then there's type 4. This is where your T cells, during that screening process, the screening process isn't 100%. Sometimes a defective T cell will get through. And if that gets through, it will cause autoimmune diseases. That's where your T cells attack your own body. And it can all hell breaks loose. Monoclonal antibodies, this was like one slide on your thing. It's also super unimportant. If you ever see a drug name that ends in MAB, it's a monoclonal antibody. If it ends in XEMAB, it means it's a rat and human monoclonal antibody. And like a drug name, if the drug name, for example, omalizumab or something. No, you cover it like in a few weeks, but next semester. If it ends in MAB, it means it's a monoclonal antibody. That's all that's important. All right? MABs are basically scientists have found antibodies that they like and that they think they can use. They've made copies of it so they can inject it into people. The two main uses for it right now is autoimmune diseases. So this is to suppress the immune system, preventing those cells from killing your self cells. And then there's cancer immunotherapy, and that's to basically to induce the immune system. So to reawake it so that they can fight the cancer cells. That is the end of immunology. We're going to take a break because I'm out of breath, and we'll start in microbiome in like 5 minutes. All right.
Great.
Alright guys, let's get started. I was going to finish. As I started making these slides, I realized that this is hell. These slides have no pictures because I don't want to really see thrush, and I don't want to see a leech or a tick or lice. So you guys will get a lot of words over the next slides. I will skip slides because you can read them yourself. You have eyes. All right. Contents. All right. Host-parasite relationships. Here are some definitions. Symbiosis basically means when one organism lives in or on another organism. Mutualism is when they both benefit. Commensalism is when one benefits whilst the other one gets nothing. And then parasitism is when one benefits while the other one is harmed. That's about it. Your normal flora. Human normal flora is mostly commensals, but there are some mutualistic ones. There are some bacteria inside your gut that literally help you absorb vitamins, help you produce vitamins, help you get rid of all the waste in your body. Okay, and so most of your normal flora are bacteria. They're acquired during birth or shortly after birth. Like as you come out, there is a lot of bacteria around there. And so you'll get all of that as your normal flora. And then throughout life, you'll get more, depending on what you encounter. The important part to remember is that your lower respiratory tract and upper urinary tract are completely sterile. If there are bacteria in there, it means something is wrong. This is a list. You can read the list. Only important thing, stomach, even though it's super acidic, there are some bacteria that live there because they're acid tolerant. All right, microbes classified into 3 domains, bacteria, archaea, and eukaryotes. Your archaea, no one cares about them because no one's found any that infect human beings yet. Prokaryotes versus eukaryotes. Surely you know this from like just general knowledge, but prokaryotes, mostly unicellular, and have no membrane-bound organelles. So if you tell me that a prokaryote has a nucleus, I'll shoot you, because they don't. Eukaryotes do. That's about it. Prokaryotes, they come in 3 shapes: cocky, bacilli, and spiral. The cocky ones all have coccus in their name, like staphylococcus, streptococcus. Bacilli, they don't come in the name, sometimes they do. But basically, round, straight, spiral, that's it. Eukaryotes, that's everything but bacteria. What they have are all the membrane-bound organelles, so they do have that mitochondria. And that mitochondria actually is the powerhouse of the cell because it does allow for the complex intracellular processes. They also do have non-membrane-bound organelles, which are important too. These are your prokaryotes. The only thing that's really of note, nucleoid. Nucleoid does not mean nucleus. Nucleoid is a region inside that cell that just literally has the genetic material. It just sits there. That part is called the nucleoid. There is nothing holding it there. There's nothing like wrapped around it. It's just there. Your cell wall. These two, no one really cares about. These two are important. I don't know if you remember, like back half an hour, lipopolysaccharides, that's what your toll-like receptors will look at. If they're shed off, if they're like released, secreted from the cell, they can cause toxic shock. That basically means death. And then there's peptidoglycan, which is what you've all probably heard about. It's that carbohydrate that's inside the cell wall, and you can stain for it. In positive ones, they're super purple because there's a lot of peptidoglycan that's on the outside, which means a stain can adhere to it really easily. Whereas on the negative ones, your peptidoglycan is on the inside of an outer membrane and on the inside of a capsule, which means that the gram staining can't adhere to it. That's why it's pink. Also on bacteria, they have flagella, so long thread-like things, and they have pili and fimbriae, which are short thread-like things. These ones help them move. These ones help them stick to things. There's one called the sex pilus. Important part, it doesn't mean that bacteria have sex with each other. It means that they can transfer DNA through a tube. Bacteria multiply by binary fission. They don't reproduce sexually. So binary fission is super simple. Basically means your DNA makes 2 copies and your cell splits. That's it. That's why it's so fast, and that's why bacteria can multiply so quickly. This slide, this part, remember it. Like literally memorize this process. Memorize it, please. Yep. The bacteria will enter the body. It'll colonize the host, which means that a lot of them. There's multiply until it there's a lot of them. It'll evade host defenses. Basically, the immune system can't get it, so it can stay. It'll keep multiplying, which I spelt wrong. Sorry. And it will also disseminate, spread throughout the body. Once bacteria spreads throughout the body, you get a systemic infection. Systemic infections are the ones that cause fevers, can cause weight loss, you cause night sweats and stuff like that. And damage to the host, blah, blah, blah. Another part that's important, virulence factors. They're basically the things that make a bacteria more dangerous. Adhesion molecules, so like your pilus and your fimbriae, they help them adhere to things. So inside your gut, all your bacteria like stick to the epithelial wall, so basically stick to your gut wall, so that as food goes through, it doesn't get pulled off, and that's how they stay there. Antiphagocytic features. So a lot of bacteria have a thing called a capsule, and it's super slimy, and it's made out of a lot of different things, but basically when the macrophage goes to engulf it, it just slips away. Last of all, toxins. That's self-explanatory. All right, viruses. If you tell me that this thing is a living organism, you're wrong. It's not. It is acellular, it is not made of a cell, and it's an obligate intracellular parasite, which means it has to live inside a cell to cause disease. This is its structure. Memorize the structure because you'll use it next year and probably forever. You have some genome in the middle, so DNA or RNA. There will be some protein sometimes, and then it's wrapped in a protein case. It's called a capsid. Then on top of that, sometimes there is a lipid envelope. On top of the lipid envelope, there can be spike projections. They basically just stick up, and they're the antigens that your body tends to detect. Classifications. You can have either RNA or DNA viruses, single-stranded or double-stranded. Really, the most important one is the HIV one, which is called a retrovirus. There we go. These are the single-stranded RNA viruses. What they do is they actually have RNA in them, but that doesn't get instantly translated into a protein. What happens is it goes from RNA back to DNA, back to RNA, and then made into a protein. That's what reverse transcription is, making RNA go back to DNA. This happens in HIV and a couple of other viruses, but HIV is the most important one. They can also be classified based on their shape, their size, what they infect, and what disease they cause, but yeah, whatever. Viral replication. Memorize this as well. Memorize the order. And super important, like the only hard part about this is it's not even hard. At the end, virus particles, also known as virions, they come out either by the cell bursting, releasing all of them, or they come out in buds, so like little bits at a time. And yeah, that's it. Viral disease, you can read this yourself, but it basically means some viruses infect one spot and then they'll move to infect another spot in the body. For example, rabies. Starts off as a bite, but it can cause CNS effects, as in brain, spinal cord, and you will seizure until you die. Parasites. These are the really gross things that no one ever likes to talk about. And the other thing is that they're not common in Australia. They're more common in animals than humans. And there are three categories. So helminths, protozoa, and ectoparasites. Ectoparasites are the easy ones. They're the ones you get as like a little child, and you get lice from each other. They don't actually cause any disease, but they can carry disease. The really gross one is meiosis. It's basically, I don't know if you've ever seen like on Facebook something where someone has literally fly larvae, like fly babies, growing underneath their skin, and it's like a pulsatile mass under their skin. Yeah, that's what fly larvae look like. Hope you never have to treat one. Helminths. They can be classified into three groups: your nematodes, your cestodes, and your trematodes. Your nematode includes things like Brugia malayi. That will cause filariasis. Filariasis is where your lymphatic vessels all get blocked, and that will cause elephantiasis. You may have seen people, probably like generally Africans, where their entire arm has swollen up so much that they look like an elephant. That is elephantiasis. It's because their lymphatic vessels are blocked and their lymph can't get back into their bloodstream, so it just stays there, and they continue swelling, and at some point they will burst. Flatworms, tapeworms. So Taenia solium, they're the ones that you'll eat if you eat undercooked meats. So from like pork, beef, etc. So make sure you cook your meat properly. And then there's flukes. If you've ever heard my 3 talk, you will have heard of schistosomiasis, and that is a type of fluke, and that comes via water. Yes.
Do you know the names or not?
Yes. I'll have a couple of slides later on where it's literally just a list of names. They're the ones that you should recognize. And the ones that are bolded are the ones that you should recognize even more. Characteristics. Have you ever seen the really terrible videos of someone pulling a tapeworm out of a human intestine, and they're super long? That's because they've grown so much, because your intestines have so much nutrients for them. Generally, they're only about centimeters, but they can be meters long. Body cavity. You don't even know they're there until they start causing symptoms. And by that time, they're massive, and they're probably eaten through part of your intestinal wall. They can attack any other organ. They can invade into your brain, and literally you start feeling your personality will change to start off. And then you realize later on when you start having that it's just a parasite in your brain that's fucked you over. All right. Transmission. All right. You can have direct transmission, which is basically when you get the egg and it goes directly into you, or you can have indirect, is where the egg has to go through an animal. So, for example, the ones I was talking about before, Taenia solium, it goes through a pig, then you eat the pig, then you get the parasite. Protozoans. These are single-celled, but they are still eukaryotes. A lot of people mix them up with bacteria, but they're not. What they do is that they infect generally via insects, or if you ingest their eggs. So insects, malaria, the most common one, you've all heard of it, trypanosomes and leishmania. Then there's infective ones. Toxoplasma, you literally get through cat feces. Ingestion, Giardia I'll spell that wrong, lamblia, that you get from, if you go to Bali or Indonesia and you eat really contaminated, dirty food or drink really contaminated water, you'll probably get Giardia, and you'll just have really bad diarrhea for an extended period of time. And then there's venereal, I can't even say it anymore, venereal transmission, basically a STI, sexually transmitted infection. Fungi. These are eukaryotic obligate aerobes, which it means that they have to have oxygen to survive, which is why your fungi never really invade your internal organs besides your lungs, because your lungs have heaps of oxygen, but everywhere else doesn't. Fungal infections will affect your skin and your lungs. That's about it. The other important thing is that 3 types, yeast or mold, and then dimorphic basically means it's both yeast and mold, depending on the situation. Yeast, they grow by literally budding off. So think exocytosis, but like outside the sun on its own. Just little buds come off, and that's new yeast cells. Whereas mold, they grow by extending hyphae. Hyphae are like these little thread things. And if the threads get tangled and form like a mat, the mat is called a mycelium. And from that mycelium, sometimes part of it will like bulge out, and that will be the fruiting body. A fruiting body is where spores can grow. In other words, that's how they reproduce and how they spread. Dimorphic, as I said, it's yeast at like a lower temperature and mold at a higher temperature, or vice versa. They're just the fancy ones. Fungal diseases, you can be allergic to them, like nuts or whatever. Same thing, you're just allergic, you get anaphylaxis. You have toxicoses, which is basically a toxin, and it will cause effects. And then you have infections. The infection and the severity of it will highly depend on whether you're healthy at the time or not. If you're immunocompromised, a fungal infection can often lead to death. Whereas if you're healthy, a fungal infection, you might not even know it's there. And then there's cutaneous or subcutaneous. Cutaneous is your skin. If it's on the skin, that's not really severe. It's quite common, but it's not severe. Whereas subcutaneous, it means that you might require surgery to get rid of it because it goes under the skin and can go into your blood, etc. All right, mycotoxins. We'll go through an example. It's called Stachybotrys. That's a family of fungi. Aspergillosis, and a couple of others come under that. And what they cause is, if you inhale it, you'll get these symptoms. So all these symptoms plus these ones here. How can you inhale them? If you've ever been to like a really old house where they have like water, like leaky water pipes or whatever, and you see like a black mold on the wall, that is Stachybotrys of some sort. And if you inhale their spores or their cells, they will basically grow inside you. And the only way to get rid of them is by activated charcoal, which is like a filter. And supportive care basically means preventing, like helping the symptoms, because you might be in pain, you might be coughing a lot, etc. So that's it. Medically important microbes. This is how you can get infections, which is pretty simple. The early one, what was I going to talk about? Nosocomial is just a fancy word for infections you get from hospitals. And they're generally the worst ones, like as in the highest mortality rate, because if you're in hospital, you're already sick. And if you get an extra infection, you're going to die. Transmission. Simply put, you can go via the air, you can go via fecal oral, which is basically someone's shit has touched your hand and you ingest it. And then there's sexually spread. It can also be spread directly, like skin to skin or like blood to blood. If you use like intravenous drug users, they tend to get a lot of infections. And then there's also, it's not on here, but you can also get infections as a baby, as a fetus, from your mother. So it being congenital, so if you're inside the placenta and the virus or whatever infection has passed through the placenta, you can get infection from that. Or it can be during childbirth or after, and via breast milk as well. That's why mothers have to be so like careful about not getting sick. On top of that, you can get infections from vectors, so insects or animals. These are definitions, learn them, like really learn them. Bacteria, this is the list I was talking about. If you remember them all, it took me like 2 years to remember them all, but the most important ones, okay, E. coli. I don't know if you heard about KFC causing salmonella back a while ago. That was because of E. coli. It caused severe diarrhea. Mycobacterium tuberculosis. You guys did a PBL on TB, right? So that should be easy. The other ones are these ones. Staphylococcus aureus and Streptococcus pyogenes. They're the two that are naturally on your skin, but when you have a defective immune system, they will cause severe harm. On top of that, Clostridium difficile. This is one of the worst nosocomial infections you can get. So in hospitals, if you get an infection of Clostridium difficile, you'll get such severe diarrhea that you basically shit yourself to death. Yeah. The ones that are really easy to remember, for example, your STIs, like gonorrhea is caused by Neisseria gonorrhea. Chlamydia is caused by chlamydia. Don't make it hard for yourself. Just remember the ones you can, and the rest of them, chuck them into like groups. Another one that's important is Pseudomonas aeruginosa. Did you guys do cystic fibrosis in PBL? Yeah. Pseudomonas aeruginosa is one of the most common infections seen in cystic fibrosis people. If the question says cystic fibrosis, just circle Pseudomonas aeruginosa and move on. Viruses. This is actually really badly classified, but I kind of just chucked them on the random titles. The important ones to remember: varicella zoster. You've all had the infection injection for it, otherwise you wouldn't be here. It's chickenpox. But if this virus goes and lies in your dorsal root ganglion, and that's basically near your spinal cord, it can come back later in life to cause shingles. By that stage, it's called herpes zoster virus. If someone tells you varicella zoster virus causes shingles, it doesn't. Herpes zoster virus causes shingles, but varicella zoster virus will lead to herpes zoster virus. And then on top of that, rotavirus, and also another one called norovirus. If the question has something about a cruise or like kids at a kindergarten, then it's rotavirus or norovirus. It's basically gastro that spreads really easily just because everyone is so unhygienic at kindergarten, as a child, and also on cruises. Fetal, the important one is probably cytomegalovirus. When infecting like a normal healthy person, it's like a flu. It doesn't cause anything. But if it infects a baby, it can cause CNS damage, brain damage, which basically, it's unfortunate if your mum's barely sick and the child comes out dead. Localized infections. They're the infections that infect one spot, and that's about it. So warts, your papillomavirus. Then there's systemic viruses. They're the ones that spread, as I talked about. Rabies, you get bitten by a dog or a rat or something. It starts off in that tissue. Just the connective tissue gets inflamed, infection, blah, blah, blah. But then if it reaches your CNS, you will get seizures. And has Paul McMenamin imitated this for you? It is creepy. He will stand there pretending to seize for 5 minutes. Next semester maybe. Chronic infections. Hepatitis B. A chronic infection is one that basically never leaves your bloodstream. HIV is one of them. Hepatitis B is one of them. If left untreated, it can lead to severe complications. So in the case of hepatitis B, liver cirrhosis. I'll tell you. Okay, so you know how there's hepatitis A, B, and C? How you get hepatitis A is if you've been in Asia. How you get hepatitis B is from bitches. In other words, you've been sleeping around. And hepatitis C, I've forgotten what it comes from, but shit. It was a really rude acronym, so I'll stop there. Oncogenic infections. They're basically viruses that could possibly lead to cancer. If you've ever had glandular fever, that's Epstein-Barr virus. And that could have led to Burkitt's lymphoma, which is basically a cancer of the immune system. Or if you have human papillomavirus, it can lead to cervical cancer. And you'll probably do an assignment on this next year. Influenza is the most important virus that I want to talk about because there are 3 types. The most important type is type A. And type A can undergo antigenic drift and antigenic shift. Please learn the definitions and how they both work. I'm running out of time, so I'm going to skip through this. But just know that influenza A is the one that causes, the panic around the world, like at the airports where they scan you with a thermometer and everything. That's swine flu, bird flu, all those animal flus. They're influenza A, and they're the ones that cause, chaos. Parasites. Once again, here's a list. Just memorize, don't memorize. Just remember T. brucei is caused by the CDC fly, while T. cruzi is by the Chagas, is Chagas disease, which is by that bug. These two, you'd rather have T. cruzi than T. brucei. T. brucei will give you CNS destruction. So basically, you might not realize it, but in a few weeks, you'll die. Leishmaniasis, generally not too bad. But if it invades your organs, if it reaches your liver and spleen, your abdomen will swell so much that it's horrendous. If you want to look up pictures, go for it, but it's gross. Giardia, I can't pronounce it. Giardia lamblia. That's the one I was talking about. If you ever see the word Bali or Indonesia in the question, circle Giardia, move on. All right. Malaria. Remember this. Falciparum. I'm pretty sure there was a question last year which literally just had 4 names. Was they're Plasmodium species. In other words, they all cause malaria. And it asked which one causes the worst. This one. Yeah. Falciparum. How it does that is because what it does is it basically causes your blood to clot. And it will clot in random spots around the body and in the vessels. If it clots, it blocks your blood vessel. It means that no blood will get through and whatever part that's been blocked off will die. If it's your brain, you're going to die within like moments. Important thing for next year or even for like HKS, how do poor people prevent themselves from getting malaria? Bed nets and insect repellent. How do rich people? We use drug prophylaxis, which is basically taking a drug to prevent malaria from infecting you. The bottom one is the gross one. Approximately 90% of French people have a permanent infection of toxoplasmosis because they eat raw meat so often. And how this actually happens is this parasite, cats ingest it, they'll poop it out. Animals eat that poop, they'll get the parasite. If you eat that animal that's undercooked, you'll get that parasite. In healthy individuals, really doesn't cause anything, but in immunocompromised people, it just in general, if they're immunocompromised and they've got an extra infection, they are in deep shit. Fungi, that stuff, read it. Oh, actually, this one's pretty cool. If there's ever a question about a rose, it will be this, Sporothrix schenki, because apparently this fungi loves roses, and if you prick yourself on a rose, it will enter your bloodstream, and that's how you get this infection. This side is the more important one, Candida. It can be simple. It can be like thrush, oral or genital thrush, which is, easily treated. But if it gets into your bloodstream in immunocompromised patients, it can cause endocarditis. Endocarditis is the inflammation of your heart. Your heart should not be inflamed. Aspergillosis, another simple one. If you've got, if you have HIV or if you are a cystic fibrosis patient, you have a higher chance of getting this. It's basically invade your lungs most commonly. It can invade other tissues, but lungs are most common. If you've had TB in the past where part of your lung has an abscess, basically a hole, aspergillosis can grow in there and form like a ball, and literally like a solid black ball. And you don't like people with the fungus ball don't realize it's there until they start coughing up blood. And then they get a massive surgery and get the ball cut out, and they're like this big, and there's just like fungus growing inside their chest. All right. Other than that, the last one, P. Jurovici. This one is pneumonia. It causes pneumonia. And the reason why it's important is because in pretty much every patient that has AIDS, they will have this type of pneumonia. Also seen in cancer patients. Strategies for control. Read this yourself. It's basically how you clean, how you disinfect, how you sterilize. Remember, wash your hands this way, that way, blah, blah, blah. This slide, we like to use radiation, but radiation takes a while. No one's going to stand there holding a UV light over a particular surface until it's done. So what they like more often is using chemicals, which we've used before, like chlorhexidine to wash your hands. Sterilization. No one really cares about the middle column. It's these two, dry and moist with pressure. Moist, great word. All right, dry, just know basically dry is like basically baking it, cooking in an oven. You can kill a certain amount of bacteria, but you also melt whatever's there generally. So it's not an effective method. What we like more often is moist with pressure. You use a machine called an autoclave, and increasing pressure means that you can increase the temperature at which water boils at, which means you can kill all those things that can survive past 100 degrees Celsius, particularly spores like fungal spores and bacterial spores. Ionizing radiation, read it. This filtration, you use it for things that can't be put into a machine. For example, like saline water. When you put saline water into a patient, you don't want it to have other stuff in it. So you filter it, as the word says, you literally put it through a filter that has holes, and you might use a pump to push the liquid and gas through it. That's it. Chemicals, there's a lot of them, but most of them are super toxic, which is why we don't use them as often. And autoclaves are generally the ones that we use the most. Oh my God, last slide. Nosocomial infections. Hospital-acquired infections, in other words. These are the ones that you get when sterilization or disinfection or cleaning doesn't work in a hospital. What can happen is self-infection. This is when a patient, like a burns victim, for example, They might have an infection on their leg. They touch it, they touch their shoulder, they touch another part of the body, and they're spreading the infection across themselves and making it worse for themselves. It's hard to control because no patient realizes that, touching their leg might actually cause death in the end. Cross-infection, that's the one that's most common, from visitors or from staff, from patients. Visitors come in and they've been walking everywhere. They don't really realize, they might give a hug to the patient and they've got an infection. And then environmental, this is probably the worst one because the hospital will get sued. What happens with that is, recently Alfred had a gastro outbreak, and they think it might be because of the food there, because all the patients get food that's being cooked at the same time, and basically everyone will be infected. And then common types, urinary tract. Anyone know why urinary tract infection is the most common? Catheters. That's literally sticking a tube up your urethra. Those tubes can be only cleaned so much. So that's an easy way for bacteria and other stuff to get in. And these are the important infections. If you can't remember any other names, remember these 5. Staph aureus, if it says methicillin-resistant Staph aureus or golden staph, MRSA, these are the ones that, surgical wounds, if they get this infection, it will literally cause the skin to, it will cause such a bad infection that you would just have to cut out that chunk of skin. And I'm pretty sure it can also lead to necrotizing fasciitis, which is basically flesh-eating bacteria. The bacteria will eat away at your flesh. E. coli, as I said, diarrhea, Pseudomonas aeruginosa, cystic fibrosis patients, and Burns patients always have infections with those. Klebsiella pneumonia, have a guess of what it causes. Pneumonia. Clostridium difficile, as I said, that's one where it's just really bad diarrhea that you get in hospitals. And that's it. Thank you. Yeah, I finished. Guys, quick thing. Because we have no time, it will be uploaded into the Google Drive. It's like a 70 slides worth of questions just on immunology and microbiome. So if you want to go through those, there we go. Guys, just another thing. The second years are now getting their pizza. They didn't take any of your pizza, so please don't take any of theirs. Second years are getting pizza. Don't take it. Simple. Yeah, sure thing.
I just missed out on the start. Are we expected to so the slide before just sort of I think summarized it which goes on the boot, that's not and also diagram. So that they differentiate into minus and then they decide. And that's just what they go. So that this comparison can move. That's very tough. Oh, very tough. Oh, right. And what's this? This is, I don't know how to tell you it's basically saying that with any T cells, they need to go through screening before they go start to work. And what that means is because they don't want autoimmunity. And so if you have a strong receptor, recognizes self-antigen really strong and thinks that it will like this offensive and I think it's a plus and then that causes negative stuff. Gets killed off the resistance. Yeah, she's about to start. Oh, you leave it off. And then there's a really good in other words, it doesn't identify self-cells as well, which is good. And so those cells stay. Okay, so that generally happens with like any defective cells or defective reflective, defective
How'd it go? Holy shit, I was like running a fucking marathon.
I am so nervous. I am
That'd be fun. I smelled you and you're like, I smell so much doing that thing. I was like, oh, you better shit yourself until you die. They probably won't.
Oh my god, you live in New York.
I know. So you know what I did? I drew one copy. This is the original. I photocopied and this is the original. Every time I drew it, so and you can't really tell unless you move. And so when I scanned it, this is
That's so nice.
so like, I have like 5 copies of it. So I gave it to Jessica because I was practicing last night just to get the right order. Because every time I draw it because I know it. I draw it. So I teach year 12 students and come outside with me. I also feel like it's like,
Oh yeah, because you ate a whole fucking pizza to yourself. Alright.
the left.
Do you reckon?
Throughout the dead slot.
So this is probably it.
You've got like, yeah, this will be most of it. I'm gonna move all my shit.
Can you grab some water?
No,
Oh, down here.
just ignore this laptop. It's just for recording.
Oh, okay.
So microphone.
Yeah, I use this one because I've moved around.
How do I do that? I'm a mover.
Pin that thing somewhere onto you.
Mouse.
This.
Yep.
Is this the thing?
Yep. And then on off, I'll leave it off for now.
Okay, is that close enough?
Yeah. They're generally really quiet. On's there. Chuck it on the table or in your pocket or something.
Oh, it's fine. Alright, I can tell you to pee.
Yeah.
No, not pee. I've already peed. I need to get water.
Yeah, is this
And
your
stop
cup?
freak- Yeah. Oh no, I've got to drink water.
What's
Lucy just
Lucy?
followed. Yeah, thank you.
And yeah, they're a nice audience.
Yeah, I get really nervous.
Yeah, I know.
When I start, like, I forgot what everything is, and I haven't practiced it, and usually it's how you can introduce yourself. I'm like, oh my god, I can't introduce myself.
Yeah, I didn't even do that. I was like, alright, let's go.
Yeah, that's me. I mean, everyone wants to go home, right?
Yeah, exactly. Is the next door, were you in the lecture?
Yeah. Yeah, but the thing is, I need to go anyway, so I'm kind of like, I really can't.
I'm not
You do that.
Yeah, I do it. Wow. I didn't touch it.
Oh, wait, when do you come back? Okay, fair enough.
I'll smile and give you moral support.
Is that Maddie?
It's fine. You'll let a teacher.
Oh, God. Does that work? Can you hear? All right, might start now because I'm sure you guys want to go home as much as I do. Hey, is this one on as well? Hello? Oh, it is. Okay. Well, I'm Emma and I'm presenting on pharmacology. So I think that's about 7 marks in your exam, or it was in ours. So it's not as big as some of the other ones you've covered today, but they examine from such a wide range of it that it's kind of good to get your head around the important bits. So, this is just a bit of credit, so okay. So I'm going to start with some definitions, then go through some of the core drugs for each, well, all of the core drugs, and some of the extra drugs for each of the different topics. So the main definitions you need to know are, so you've done your pharmacokinetics and pharmacodynamic lectures. I know they sort of got muddled up for you guys, I think, but I hope you've eventually got them. So the things you need to know about that is that there are different ways you could administer drugs, and that'll depend on whether you want people to be compliant or whether you want them to get to the source quickly, whether people are able to actually take an oral drug or whether you're going to need to inject it for some other reason. So the main ones you need to know are oral, sublingual, so under the tongue, which goes directly into the vena cava. Rectally is extremely rapid, or else injection is obviously has the greatest bioavailability, which I'll be talking about in a moment. Something to know is the pharmacokinetic order. So the thing ADME is the best one. It has to be absorbed from the area of administration to the bloodstream, then distributed, metabolized, and then excreted. So that's pharmacokinetics, which is sort of what your body does to the drug. And then you've got pharmacodynamics, which is what the drug does to the body. So when it binds to a receptor, what sort of action it's going to have. So these are the three ones that they'll often ask you to define. So there's affinity, efficacy, and potency. So affinity is the actual ability of the drug to bind at the receptor site, whereas efficacy is its ability to bind at the site and then have a biological effect. So I don't know if I go into it. I'm going to assume I go into it. And then potency is the last one, which is how much of the drug you need to give in order to produce a given effect. So if you've got a drug with a higher potency, that will mean at a lower, you'll get a response at a lower concentration than a drug with a lower potency. The other terms you need to know are agonist and partial agonist. So earlier I was talking about the difference between affinity and efficacy. So a drug which has both affinity and efficacy is called an agonist, whereas a partial agonist, it also has affinity, but it's going to have less efficacy than a full agonist. So I'll be showing you a diagram later, and you'll be able to see the difference between that. An antagonist is the opposite. It has affinity, it can bind to the receptor in the same way, but it can't have any actual effect on the receptor. So it usually blocks the actual, like the desired effect from happening, which can also be what you're looking for pharmacologically occasionally. Finally, there's inverse agonist. So that's the complete opposite. So it binds to the site, but it actually has the opposite effect to what the agonist would have. Yeah, so on this one, I've covered them up. I was hoping some people might be able to guess which one would be which. So there's the 1, 2, 3, 4. Would anyone be able to tell me which one the top would be? Yeah, and then down the next one. And the one along the line? Yeah, and then the one that's doing the inverse effect, that one's going to be the inverse agonist. Yeah? I think these are the last two definitions. So first pass metabolism is the fact that when you get a drug into your circulation, when it's metabolized, you're going to reduce a lot of the actual availability that you've got. So before it can reach your systemic circulation, when you're going through the ADME phase, you know, the absorb, distribution, metabolism, etc., it actually can lose a lot of its concentration there. And bioavailability is something to know. It's a measure of how much a drug is absorbed into the circulation over time compared with IV administration. So I've got this diagram here, which I drew, but you can probably tell. And so IV is this one here. It starts at 100%. It's 100% available in the bloodstream, and then it goes down from there. Whereas this one here, this could be orally, it could be rectally, it could be any other form of the drug. And the most important thing to see is that it never actually reaches 100%. So it never reaches quite the same levels as IV. So it will never have the same quite amount of response. Oh, no, this is the last thing. I wasn't sure if this was from the first sem or if we did it a bit later, but minimum inhibitory bacteria concentration is the minimum concentration that will stop bacteria growth. They're kind of self-explanatory from the name. So inhibitory, it inhibits growth, but it doesn't actually like reverse it. Bactericidal kind of sounds like suicidal, so it's about killing the bacteria and it reduces the number. And then the minimum effective plasma concentration. So as I said, they're quite self-explanatory. So this one is literally just the minimum amount of the concentration of the drug that you would need in the plasma to have the therapeutic effect. Oh, I keep thinking I'm done with these definitions. Time dependent. So these ones, there was a good graph for it, but I couldn't find it. These ones are only effective for the time in which they're above a certain concentration. So if this is the concentration you need, the drug goes up and it stays above that for however long, that's how long it's effective for. Concentration-dependent drugs will be more effective if they're above that level for longer. So if they're further above that level. So the more concentration you've got, the more of an effect you're going to have. Whereas time-dependent, it doesn't matter how far above that minimum concentration it is, as it's about how long it's above that concentration for. So I think I have two questions here. So what are the phases of pharmacokinetics? Anyone? Yeah? D, you're going to do a bit late. It's really hard to hear. Yeah, so D. And then, okay, so these are the drugs. These are the orders I'm going to go into. So cancer, antibiotics, antiviral, antifungal, antiparasitic, and then neuromuscular drugs and antivenoms, which is that module you would have done, the online ones still? Yeah, thanks. Okay, so starting with cancer treatment. So these are the major drug classes that you need to know. So cytotoxic, These ones stop the process of transcription and translation. So a lot of them will actually have the same effects as some of the antibiotics, but they're too toxic for infectious use. So not only do they hurt the cancer cells, but they'll also hurt some of the just regular body cells. But in cancer, because you've got that different cost versus benefit ratio, you can still use them even though they can harm off some of your own cells. Alkylating agents insert an alkyl group into the DNA structure, and that forms covalent bonds, which sort of joins up the two strands of DNA, and so they can't be pulled apart. And as you probably know, the DNA pulling apart is one of the first steps of it being able to be replicated, so this stops it from being replicated. And antimetabolites, they substitute themselves into DNA synthesis and block it. So essentially, they'll get incorporated into the DNA strand, but they haven't got whatever's needed at the bottom to attach on another one of those. So no more synthesis can occur. You can't add any more, I can't remember what they're called, beginning with P, the things that get added on. And then hormone antagonists, they act against cancers which require sex hormones. So they'll act instead of the hormones, pretty much. I'll go through them when you get an example of each of them. So these are the ones that are core, I think. In fact, I think one or two of them might not be core, but they're all the important ones that were mentioned in the lectures. So 5-fluorouracil is one of the antimetabolites. So you might remember that was the one that would insert itself into the DNA synthesis and stop it from occurring. So 5-fluorouracil is a pyrimidine antagonist. So basically it'll be misincorporated into the DNA. And because it's affecting the DNA synthesis, it's S-phase specific. The way I was taught to remember that is that a 5 looks like an S, so it's S-phase specific. The next one, methotrexate. So this is one of the ones I was talking about that is another antimetabolite, but this one's a folate antagonist. So since folate is required for purine synthesis, DNA synthesis is also disrupted, and once again, S-phase specific. Some of the ones to remember, meth isn't pure, I read somewhere, so it mucks up purine synthesis, and meth is also awful for babies, so it's not for pregnant women. So that's some of the adverse effects, not for pregnant women, and it can also have neurotoxicity and bone marrow suppression. I'll talk more about bone marrow suppression and why it does that in a second. I'm pretty sure. Yeah, peek down here. Cyclophosphamide is an alkylating agent, which is one of the ones that cross-links between adjacent DNA strands. This one is Cyclops are Greek, so Greek G guanine analog. So it's going to replace itself between adjacent guanine bases and then cause that, it's unable to pull apart and you get apoptosis. This can occur is cycle non-specific though. So what I was saying earlier about bone marrow suppression, hair loss, infertility, all of these ones, it's about the fact that it targets cells which have a quick turnover, like cancer cells, because that's one of the few things that make cancer cells special that they can sort of attack. But unfortunately, we have some regular cells which also have a very fast turnover. Someone said that cyclones spin quickly, so that's why you have the fast turnover cells that get affected in your adverse effects. Doxorubicin is one of those cytotoxic ones. So one of those ones I was saying is sometimes used as an antibiotic but also will harm human cells. So it has 3 main methods of action. So it inhibits transcription and translation. It binds to topoisomerase and it generates ROS, reactive oxygen species, which damage the DNA. And once again, for most of the anti-cancer drugs, you're going to have pretty similar effects. So it's the hair loss, it's the nausea, vomiting, which I'm sure you've heard about from chemotherapy, and the targeting almost all cells with rapid replication. Something to know about this one is that it's metabolized by CYP3A4. You don't need to know that necessarily, but the fact that it has drug interactions means it'll alter the, it will mean that it can have more drug interactions with other drugs. Tamoxifen is one of the hormone antagonists. So there's 2 of them. There's tamoxifen and anastrozole, which are 2 hormone antagonists. Tammy and Anna are both girls' names, so these ones act on breast cancer. So this one is a selective estrogen receptor modulator. So it acts on the estrogen receptors, which are on the breast cancer cells, and they need that to grow. So when estrogen comes and acts on them, that sort of stimulates them to grow further. This one is an antagonist. So it sits in those spaces, has no effect, and is basically prevents them from replicating. The important thing to note is it's an antagonist in breast tissue receptors, but an agonist of bone. So it's not going to affect bone in the same way that it's going to affect these cancer cells. And that it only suppresses breast cancers which are estrogen dependent, which is only a small number, and they actually do have to test whether it's going to be effective at all. Because it's having this effect on estrogen, it's going to have similar symptoms to menopause. So this is the hot flushes, the sweating, just everything else you've really heard about linked to menopause, I suppose. Anastrozole is the other one I was saying, another hormone antagonist for breast cancer, an anastrozole one. This one's a non-selective aromatase inhibitor, which is only used in postmenopausal women. Someone once said that stro sounds like strobe lights, which were big in the 70s, which are like for old people. So it's only for people who are over. Usually the question will be quite specific. It'll be people who are 70, 60, 70, like clearly in that age group. So basically what it does is it reduces the level of all circulating estrogen, which means that estrogen-dependent breast cancers can't grow. Oh, and then there's a couple of non-core ones, which you would have had in your, not in your pharmacology lectures, but I thought I'd include them anyway. They were in the anti-cancer lectures. So one of them is for prostate cancer. It was I can never say this, gonadotropin-releasing hormone analog. So what it does is it initially causes an increase in these two hormones, in LH and FSH, because it acts on those receptors and causes their release. But then you've got this negative feedback loop. So if there's more in the blood, that's going to mean that it's going to actually stop it from being released. So what happens is they sort of work this bulb. They give it at like set intervals every so often, and overall that will decrease the levels of LH and FSH, which you need for some cancers. And this one, trastuzumab, is the MAB means it's a monoclonal antibody. So this one binds to extracellular HER2 receptors to prevent activation of intracellular tyrosine kinases. I've seen quite a few really good ways of remembering this one, but I couldn't actually find one when I was doing this PowerPoint. But it's for HER2, you'll be able to find one. I just can't think of it right now. Okay, so some questions, if you can. Which should you not give to pregnant women for teratogenic? Just means harm to the child. What to do? Pardon? Yeah, D, so it's methotrexate, and the other ones should be okay. And which of the following would be most suitable to administer to a 75-year-old woman? Yeah, I think I had enough results, so that's the one for that. Which of the following is an S-stage specific anti-cancer medication? Anyone? Yep, I can't actually see A. Yep, I've heard A, so that's methotrexate. Yep, that's right. Okay, so going on to the antibiotics. So these are the ones that are actually core drugs, so you do need to know at least a little bit about each of them. I've tried to highlight mainly the key facts. So the first one is obviously amoxicillin, and that's a beta-lactam antibiotic, and it inhibits the transpeptidase enzyme irreversibly. So basically, transpeptidase will normally act between different polymers in the cell wall and connect them, which holds it nice and strong. When you've got penicillin, it's going to inhibit these from working. So the cell will become a lot weaker, and then it can become, then it can lyse. So this is the first-line drug. It's probably, the fact that you've probably all heard of it, it's why it's the first-line drug, and it's mainly for gram-positive bacteria. In cells without a peptidoglycan wall, so this is things like Mycobacterium tuberculosis, it is which has an impermeable cell wall, it's far less effective. The main adverse effects, there aren't that many unless you've got allergies, and anaphylaxis are the two main ones, as well as also resistance, which isn't really an adverse effect, but is a bit of a problem. I don't know if she gave you the policeman example, but it's renally excreted. I don't know if she gives that example of like someone peeing it out and then they're reusing it when they were first inventing penicillin. That's her way of reminding you that it's a renally excreted drug is the thing to get from that. Cephalexin is pretty much the same thing. It also is a transpeptidase inhibitor. It's also renally excreted. It has the same problem of resistance. It's slightly more toxic, which is why it isn't the first line drug. It's got the issue of neurotoxicity. But when you've got a penicillin sensitive patient, it will be the first line for them. Something else to note is that augmentin is a name that they might put in the exam. That means penicillin with clavulanic acid. So I'm not sure if you remember what clavulanic acid. Yeah? I'm seeing a few nods, just anyone else. It's so as I was saying, penicillin is a beta-lactam. So what bacteria will do to try and stop penicillin from working, to sort of override it, is to produce beta-lactamase enzymes. And what they do is they break down that beta-lactam ring and they stop penicillin from working. Clavulanic acid it inhibits those enzymes, so it stops penicillin from being broken down. And that's what that is. And yeah, so that can be given with cephalexin, but also penicillin is the main one. And this is that brand name for it. Vancomycin is the one that you're supposed to remember. You're supposed to remember that it's like a little red van. So red van, little red man. It's the big thing to know about this one is that it causes red man syndrome because you'll get histamine release, which will cause vasodilation. And basically it'll mean you'll turn really red is the important thing to know about it. If you think of an ambulance, it's got that plus on it. So that's the way to remember that it is for gram-positive bacteria. This one inhibits transglycosidases. So just like transpeptidases cross-links polymers, this one cross-links glucose in the cell wall. So it's going to have that similar effect, but the cell wall is not going to be as strong and things are going to be able to get through it and the bacteria will also lyse. It's a far less used drug because of the side effects. So it's only used for really severe infections or where resistance is a problem. So MRSA, it stands for multi-resistant Staph aureus, and that's one of the cases when vancomycin would be used as a third-line drug. Chloramphenicol, couldn't find the class. This one irreversibly binds to the 50S subunit. So there's going to be like about 4 or 5 drugs, which I'll talk about, which bind to one of the subunits. So as you probably remember, when you're replicating DNA, you need the RNA to act. You need the RNA to go through the ribosome, which has 2 parts. It has the 50S subunit and the 30S subunit that have to come together. If something's binding to them, they can't work, and that's the basis of the next probably 4 drugs. So chloramphenicol irreversibly binds to the 50S subunit, and the main thing to know about this one is that it causes grey baby syndrome. That's sort of the buzzword that will come up. If you drop a baby in a chlorine pool, it'll go grey is kind of the way to remember that, and it works. Erythromycin is a macrolide. There's 2 of them like this. So the difference between erythromycin and the one I just spoke about, both for the 50S subunit, know which one's reversible and which one's irreversible. So this one's reversible, as is the next one, which I think is clarithromycin. It's a second-line antibiotic. Once again, for penicillin-sensitive patients, it'll become the choice drug. It's also mainly used for upper respiratory tract infections. So erythromycin has throat in it, throat, there's got to be something there. And once I was just saying clarithromycin and erythromycin are the same thing. As in the same method of action. I don't know why I put this one next. I'll come back to that one. Doxycycline is another one of those ones that binds to the subunit. This one reversibly binds to the 30S subunit. That's pretty much all I can say about it. It's the side effect to know about this one is that it causes calcium deposits in teeth and bone, so it can suppress bone growth and it can cause yellow teeth. So it's generally not given to children who are still growing quite rapidly and who need good bones. So it's not given to children under 8 or 10. It's hard to know if it's 8 or 10, but if it was in an exam, it would be something like a 4-year-old child. It'd be quite obvious. Or pregnant women. The way to remember that is that cyclists need good bones and it ruins your bones. The other one that is the subunit one is gentamicin. So you've got erythromycin, which is 50S reversible, chloramphenicol 50S irreversible, doxycycline reversible 30, and then finally irreversible 30. So there's sort of one of each. Gentamicin is the irreversible 30S subunit, and you can remember that because a gentleman never leaves his lady unattended, so irreversible and also 30S because, I don't know, creepy man with a young lady. It's a broad spectrum antibiotic and it's used for serious gram-negative. No, you're putting me off. It's used for serious gram-negative infections. It's also used for systemic infections and sometimes it's used prophylactically as well. But the adverse effects, if you think of someone who is old, they're more likely to be unable to hear or to have kidney problems, so nephrotoxicity, ototoxicity. What happened at the bottom? Okay. I don't think any of those are that important, but if you want to read up on them, they're just some points to know. Didn't know any of them. Didn't even know where I got that from. Ciprofloxacin, back to this one. This is I don't want to say that. It inhibits topoisomerase, which stops DNA synthesis. So basically, it prevents that supercoiling of DNA, which is necessary to commence replication. So if that can't happen, DNA can't replicate, and you're going to have the bacteria won't be able to spread. Pro is like a pro athlete. So if you're a pro athlete, you're going to have more likely to have cartilage and tendon damage, which is why these are 2 of the side effects. Phototoxicity is another side effect, which means sensitivity to light. There's another one of those up there. It's a pretty broad spectrum antibiotic. Something to note, it's metabolized hepatically, so in the liver, and so are a lot of drugs, which is why ciprofloxacin will have a lot of drug interactions. It's pretty vague, but just, no. I can't really think of any examples, just it can have interactions is probably the thing to know. Metronidazole is another one. So this one is taken up only by anaerobic organisms, which can turn it into a secondary product, like an intermediate product, which is quite unstable and will actually destroy the bacteria from inside. So the intermediate, it interacts with DNA and it'll inhibit nucleic acid synthesis. The adverse effects, the metallic taste. Okay, so it sort of comes down to the metro bit, like you can't drink on trains, but also think of trains. Metallic taste, and you can't have alcohol interactions because you shouldn't drink on the train. The azole thing I found the other day. So azole sounds like arsehole, and you can't breathe out your air out your arsehole. So it's only for anaerobic bacteria. And if that works for you, great. A couple more today, rifampicin, and I'll go on to isoniazid in a second. So rifampicin is one of the ones you should know as a TB drug. So it inhibits the RNA polymerase enzyme and will be used for TB treatment in combination, only in combination when you're treating TB because it's so resistant, and can also occasionally be used for Staph aureus infections. The main notes that I was just saying, it's always used in combination because TB can get so resistant to it. This is the one that it's used in combination with. And from what I remember, the actual mechanism wasn't known that well, but to know that it works against tuberculosis is important, and it does something to inhibit mycolic acid. And that's just all you really need to know about it. So some of those questions. Which of the following does not work by binding to either the 30S or 50S subunit? Yeah, yes, C. Which of the following should not be administered to children under 8? I heard some C's, but maybe not. D, doxycycline, which is the one that can cause the calcium deposits and also shouldn't be given to pregnant women. Which of the following is responsible for grey baby syndrome? Cool. Chloramphenicol. Basically, of this one, which one causes red man syndrome? Yeah. And which of the following is the TB combination? Yep. There's actually two more. There's another one. I think the thing is ripe. So if it ever comes up, it's ripe. It's rifampicin, isoniazid, and then one beginning with P and one beginning with E. I should know. Ethambutol and P. Pyrazidamide. Fantastic. No idea. Okay, this is going faster than I thought, but antivirals next. So I put 3 of them down, but actually there's a couple more in the PowerPoint as well that aren't here. So the first one is acyclovir. So the thing about antibacterials is they're far less specific. Like they can be used for a wide range of bacteria. Maybe they can only be used for gram-positive or gram-negative, but essentially they've got a much broader like spectrum. Whereas the antivirals tend to be for just 1 or 2 viruses, and that's where they'll usually get their questions from. So acyclovir. This one's used for herpes simplex and shingles, and that's pretty much it. It's a guanine analog, which, as I was talking about with the anti-cancer drugs, it competes with other nucleotides, gets incorporated into DNA, and DNA synthesis will stop. It prevents elongation. It has GI disturbances and sometimes a local reaction at the site of injection site, but that's not too important. Amantadine, I don't think this one's core, but it's still quite an important one. So the way to remember this one is that Amanda won't take off her coat. So it stops the protein from uncoating, which is one of the essential steps in, making more. Method of action. This is the specific method of action if you want to know it. Otherwise, just stops virus uncoating will be enough. Interferon alpha. So this one's used for hepatitis B and C, and I remember this one because it's like ABC, like the alphabet. So interferon A acts on hepatitis B and C. It's an immunomodulating cytokine. And so basically what that means is it helps to bring in more of the immune cells, which will help fight the reaction. So it basically improves your immunity, your active immunity. Oseltamivir is used for influenza A and B. This one's the same thing, blood types A, B, and O. Oseltamivir begins with an O, and then it is used against influenza A and B. It's a neuraminidase inhibitor, which basically means that it cleaves the virus cells, like takes them off the cell surface. So normally they have to come to the cell surface and then they can be released into the bloodstream. But what the neuraminidase inhibitor does is it actually pretty much cuts it away from the surface so it can't leave the cell. If it can't leave the cell, then it's not going to be able to spread. The thing about that one is it's virus static. So just like I was saying, there's bacteria static and bactericidal. It's the same with viruses. So it doesn't actually kill the virus cell. It just stops it from making more. So it's inhibiting the growth of the virus. Oh, these are the antiretrovirals. So the main retrovirus to know at the moment is HIV, which is the one I'm mostly speaking about here. So ritonavir is, there's ritonavir and there's zidovudine, which I'll be talking about. Ritonavir is, as I said, used for HIV, and this one binds to viral protease and inhibits its action. So the viral protease will sort of cut up the proteins and, polyprotein cleavage. It will cut up the proteins, which allows the proteins to then mature, and if they can't mature, then they're not going to be mature. I don't know why I said that. It has a lot of interactions because of, the one thing about the CYP enzymes, again, you don't necessarily know which one it is, but it's got interactions with enzymes in the liver. Doritos don't have much protein is the way to remember this one. So ritonavir, and if it doesn't have much protein, that's because the protein isn't working. They're not making them properly. Whereas zidovudine, this is a more, it's called an NRTI, nucleoside reverse transcriptase inhibitor. It gets incorporated into DNA, so transcription is terminated. But it can have GI disturbances. I always used to remember that Zido sounds like zero, so there'll be Zido problem for your baby. So this one's the one that you can give to someone who is pregnant to prevent vertical transmission, so from the mum to the baby. It's sort of like a prophylaxis for the baby. Okay, I've forgotten the answer to this one. Which drug acts as a guanine analog? I think. Oh, that's from you. I think it's a cyclovir. Yeah, cool. And which of the following can be prescribed to a pregnant woman? Yeah. Okay, the antifungals. I think there are, oh God, there are more than I thought. Okay, fluconazole is the one that we got. I sort of put in this diagram from your lecture to try and help that one along. So you've got this whole process where you've got the acetyl-CoA, it becomes cholesterol, and it goes on. And eventually what happens is it goes from here to here, and that's what goes into the cell membrane. If the enzyme is inhibited, or if any of these enzymes along here are inhibited, you're not going to be able to make the next product. And this is the product that you need in order to make the membrane quite strong and unpermeable. So fluconazole inhibits this. It's the 14-alpha demethylase enzyme, and it's needed for ergosterol production. So without that, the cell wall's integrity is lost. Amphotericin. Earlier I was saying that ergosterol is the one that's part of the cell membrane. Well, amphotericin binds to ergosterol irreversibly. And what that means is it'll create these pores in the cell membrane, and that will change its permeability. And basically with all of these, if the permeability of the wall decreases, the cell loses its integrity and will die. So this one is, the interesting thing about this one is fungicidal, virucidal, bactericidal, all the same thing. This one's only fungicidal in higher doses. So at low doses, it's just fungistatic. This is more extra information. I tried to include everything, so if you wanted to look up more, you could come back and look through these slides. I'm pretty sure they've got most of the details you'll need to know from ePharm, but don't necessarily need to say them all. I don't think they're as important. Nystatin. Okay, so this is almost the same as amphotericin, except this one's used for topical infections. So skin infections, candida, vaginal infections, GIT infections, not GIT infections, don't know why that's there. You can put this on your skin for these ones. And it's pretty much the same composition, same effect, but just different administration. Okay, I didn't have any questions on this one. Has anyone got any questions so far? Okay, cool. This one's going back to the neurotransmitter unit. What's the time? Have I got? Yeah, okay, plenty of time. This is going back to that unit that you would have done online. So there are about 4 core drugs for this, which each acted on a, which each act in a different way. But you've also got this, which is the most important thing I think to have taken from those online lectures. So, well, one thing to know is it wasn't pharmacology, so I didn't include it, but you should probably go back and look at the different, like the fact that sympathetic and parasympathetic have different length of their ganglions. I don't really know what I'm talking about right now, but I know it's important. It's like how a postsympathetic synapse is near the organ, that stuff. That's one of the important things from these modules. The other important thing is this. So these are the symptoms of muscarinic and nicotinic agonists. So agonist, I'm sure you remember this, but it's when something acts on it and has an effect at the receptor. So it's a positive effect rather than the antagonist. So the muscarinic ones you can remember is sludge. So these are the main ones you'll get. So if something agonizes the muscarinic receptors, you're going to get salivation, lacrimation, urination, defecation, GI cramping, and emesis. So these are the muscarinic ones. And nicotinic is Monday, Tuesday, Wednesday, Thursday, Friday. So muscle cramps, tachycardia, weakness, twitching, and fasciculation. Yeah, I sort of see, no? Emesis is vomiting. And the other one? It's like, yeah, it's kind of like twitching, but it's a similar thing. Anyone else? God, that's off-putting. I feel that's so off-putting. I have such sympathy for lecturers. Okay, these are the drugs. Okay, there's 4 of them that you should probably know. Pyridostigmine is the first one. It's an anticholinesterase. So when you've got in the neuromuscular junction, so acetylcholine is the main autonomic. So acetylcholine is used for parasympathetic and somatic messages. So what happens is that'll go into the neuromuscular junction, act on the receptors and have an effect. Cholinesterase breaks down the acetylcholine, so it can't work anymore. So pyridostigmine is an anticholinesterase, so it stops the thing that breaks it down and stops it from working. So basically, it'll mean you'll have more acetylcholine for longer. It stops the breakdown. It is non-specific, so it will work at the muscarinic and the nicotinic receptors, which means it's going to work for the parasympathetic as well as the somatic. The important thing about this one is that it's used to treat myasthenia gravis. That's an autoimmune condition, which you'll learn about later, but the most important thing about this one is the ACH receptors are blocked by antibodies. So you're going to need more ACH. They're competitively reversibly blocked. So if you've got more acetylcholine, it can start to go into those receptors. So basically, if you're getting more into that neuromuscular junction by using pyridostigmine, it's going to be able to knock out the antibodies and help this condition be resolved slightly. However, I said that it acted on nicotinic and muscarinic receptors. The one that you want is the nicotinic effect, which is the somatic nervous system. So atropine is used as an antagonist of muscarinic receptors. So basically, if you have this agonist, the pyridostigmine, it's going to be causing your nicotinic effects, but also the muscarinic ones. The muscarinic are all those sludge ones, which are unpleasant is mainly the thing. So if you've got a competitive reversible antagonist of muscarinic receptors, it will stop those sludge effects from occurring. So that's why it's used in combination with pyridostigmine. So you get the nicotinic effect without all those side effects. Rocuronium is a non-depolarizing muscle relaxant. So there's 2 relaxants to know about, the ones that depolarize the junction and the ones that don't. This one doesn't depolarize it. It sits in the receptor and stops anything else from acting on it. So you still can't get any effect because nothing can act on those receptors, but it is not actually depolarizing the receptor itself. This acts on nicotinic receptors and it blocks the acetylcholine receptors so that acetylcholine can't bind to the receptor sites. However, like I was saying earlier, because it's a competitive one, just like those antibodies were competitive, if you keep adding more acetylcholine, you can override this effect. Succinylcholine is the other one, and this is the depolarizing one. And this one works on the theory that once you've depolarized that receptor, it can't repolarize for a while. So if you have something that sits in there and depolarizes it, it's going to cause muscle relaxation and paralysis because it can't repolarize to transmit more action potentials. So it actually has an action that's similar to acetylcholine, but because it stays in the reception longer, it breaks that action potential cycle, so it can't be replenished so easily. But just like acetylcholine, it will be broken down by cholinesterases, but slower. This is the one that's used for tracheal incubation. That's those buzzword to know is succinylcholine is for tracheal incubation. It can also cause muscle pain and bradycardia, which are those opposite effects of nicotinic agonism. So they're the antagonist effects, because as I was saying, nicotinic causes tachycardia, so an antagonist is going to cause bradycardia. Couple of questions and then one more section and then you can go home early because I've spoken way too fast. So which of the following is not a side effect of excess ACH on nicotinic receptors? I had C, but E? Yeah. So nicotinic receptors is the Monday, Tuesday, Wednesday, Thursday, Friday. So the other one that's missing there is the tachycardia. So you will get the emesis, which is the sludge effect. And which of the following is used for tracheal incubation? If I can teach you one thing from this section, it'd be this. Yeah? Succinylcholine? I think, go ahead. Yeah. And finally, which would you give for a patient with myasthenia gravis? Just show you something. Anyone? I had A, which is sort of correct, but not the most correct answer. D? Yeah. So periodistic mean with atropine to override those effects. This is the absolute last slide. Okay, it's about snake toxins. I don't remember if you remember that one. Would have been one of your first pharm lectures. So the snake venom, it's mainly protein, but it also contains some neurotoxins, which can either act pre-synaptically or post-synaptically, depending. So they act on the nicotinic acetylcholine receptors, and there are 2 of them. So the post-synaptic, I remember that alpha is after. So the alpha neurotoxins act post-synaptically. These ones you can only it says only responds to neurotoxins. I'll change that. It only responds to treatment if it's given really quickly for those alpha postsynaptic ones. The beta neurotoxins, beta before, are for presynaptic. So they're rapid and reversible, and they respond to both anticholinesterase and antivenom. But the main thing is just alpha is postsynaptic and beta is presynaptic. And that's it. So thank you so much. If you have any questions, feel free to email or Facebook me or whatever. Okay.
It's all cold by now.
What time is it? I think I was supposed to go to 5, and I started late.
No,
Half an hour.
I need. Is that your USB?
Yeah, the red one.
I have no idea how to get USBs out, so I'm just going to go right out. So
I'm not injecting.
there you go. Normally I try, but there was nothing.
Very helpful.